Alright, hello everyone. Here we are for another 328 video, and I am actually in a different room this time, so hopefully I have it set up properly so you can hear me well and you can see the slides. One suggestion that I would have though is maybe as you watch this video, maybe open the PowerPoint and then have that in the background so that you can see some of the finer detail of the slides that you might not be able to pick up on the video. So hopefully you can hear me well enough to follow along. Now this is going to be the extra credit assignment. So you're going to watch this video and then you'll answer some of the kind of a form that I'm going to give you, and it'll be 8 points of extra credit. But this information will also help you on upcoming exams. So if you get this information down, it'll not only help you with the extra credit, but it'll also help you with the exams. And so obviously this will be part of study guides in the future and everything. And I think this video hopefully will only take about 30 or 40 minutes. So we'll go ahead and get through this video. Now the first part of this lecture actually has to do with atherosclerosis, its prevalence, and so how common it is, and then we'll go through the steps of atherosclerosis. And then the last part of this lecture will involve how diet affects lipoproteins, and then how those lipoproteins would be involved in atherosclerosis. So this is kind of where the rubber meets the road as far as nutrition and atherosclerosis. So kind of keep that in mind as we go through this. Alright, so let's get started. Hopefully I can see my notes okay. Now, the prevalence of atherosclerosis. Now, heart disease or atherosclerosis is basically the #1 killer in the Western world. So it's extremely common. And so what we're going to see is what are some factors that are involved in whether or not an individual is going to be likely to develop heart disease. The description of atherosclerosis. Now, atherosclerosis is basically, it involves the accumulation of cholesterol or lipids in arteries. And then that artery would tend to narrow because of that. And then we have an abnormal formation or an abnormal surface on the inside of that artery. Now what we're going to see is that sometimes that process, that accumulation of lipids and cells, can actually cause the blockage of an artery. If that artery happens to be a coronary artery, then that's obviously problematic because that can lead to lack of oxygen delivery to that cardiac tissue, nutrient delivery, and that can actually kill a part of that heart tissue. And if it's severe enough, it can actually cause the entire heart to stop beating and functioning. So the other thing that we're going to see from this though is that there are a lot of other molecules that are involved in that process. So I would say the description of atherosclerosis is the accumulation of cholesterol or lipids in the arterial wall and the blocking of an artery due to that collection of lipids. Now, the main risk factors. Now, I would say the main risk factor here is genetics. So hopefully, we've mentioned this before, but choose your parents wisely. Here's another example of why you did that. I'd say genetics is the #1 factor. So in other words, if atherosclerosis tends to run in a family, then that's the #1 risk factor. The other risk factors that I have here are kind of in no particular order. But let's talk about male gender then. Now, women have higher HDL levels. And because they have higher HDL levels, that tends to provide enough of a protective effect that women basically live a few years longer than men because of that. So that HDL, remember, its function is to basically help clear extra cholesterol from tissues and take it back to the liver for possible excretion. All right, cigarette smoking. Now, cigarette smoking can be a very, very significant risk factor for atherosclerosis or heart disease. It's the main cause in about 20% of cardiovascular disease deaths. So that's really significant. Now, among other things that cigarette smoke does is cigarette smoke tends to make the blood more likely to clot. And we'll see why that's important as we come up on this. We'll see how blood clotting is involved in atherosclerosis, and then you can tie in the smoking to that as well. Now, another thing that smoking does, though, is smoking tends to oxidize molecules in the blood or the cells lining the blood vessels. Okay, so that oxidizing of those molecules in the blood, for example, LDL, can be oxidized by molecules within the cigarette smoke, and that can alter LDL particles or modify LDL particles. The other thing that it can do is that smoke can actually damage the cells that line the arteries, the endothelial cells. So we'll see how that can directly lead to atherosclerosis coming up. What else? High blood pressure. That increased pressure within the arteries can actually damage the cells that line the arteries. And we'll talk about how that's involved. So one of the reasons why it's so critical that a person maintains normal blood pressure is to decrease the risk of atherosclerosis. So today we're going to learn about how all that comes together. Obesity is another significant risk factor for the development of heart disease, especially if there has been adipose tissue or fat accumulation around the waist. And that can sometimes be the typical site of weight gain. It can be a main reason for increases in blood LDL. One of the things that it also can lead to is insulin resistance. In other words, the more adipose tissue a person has, the less sensitive their tissues are to insulin. And because of that, blood glucose tends to stay higher because the insulin is no longer effective in getting that blood into the tissues. That glucose into the tissues, so blood glucose stays higher. One of the things that does is that high blood glucose can interact with proteins and molecules in the blood, including LDL. And it's actually that interaction of glucose with LDL that can also alter LDL. And we're going to see that altered LDL is more likely to become a problem for atherosclerosis. Now remember, the insulin also is a factor. If that insulin increases in the bloodstream, it can also drive up cholesterol synthesis in the liver. We talked about that before when we talked about insulin upregulating HMG-CoA reductase. Okay, so obesity can cause quite a few different problems, including insulin sensitivity, and then that can lead to some of the other problems that I just mentioned. Now, sedentary lifestyle. You can sometimes put these two things together as far as sedentary lifestyle and obesity. One of the commonalities there is that sedentary lifestyle tends to decrease insulin sensitivity. And as soon as a person starts exercising or performing any kind of physical activities, their tissue tends to be more sensitive to insulin, which means the insulin would be more effective in dropping blood glucose, and then you would avoid some of the things that I just mentioned. All right, main biochemical risk factors. Things like a high plasma or blood LDL cholesterol. It's an obvious risk factor. Low plasma HDL, because you lose that protective effect if plasma HDL is low. High plasma triglycerides are also an independent risk factor. So any of those three items there, they would all fall into the category of, lipids in the blood. And if LDL or triglycerides are high, that leads to increased risk for heart disease. And then we also have diabetes. Diabetes is a really significant risk factor for atherosclerosis, and it has to do with a few things. For example, diabetic individuals tend to have higher blood triglycerides, and they also a lot of times have lost their sensitivity to insulin, and so their blood glucose might tend to be higher, and then that blood glucose can interact with LDL, and those are a couple different specific items of how diabetes can actually lead to heart disease. Now, it doesn't mean that a diabetic individual cannot, do everything that they can in order to maintain normal blood glucose levels, and then that individual can decrease their risk of developing atherosclerosis. But it's extremely important for a diabetic to try to treat blood glucose levels so that they do avoid this risk factor. All right. Now, the information that we're going to get into now is based on this, what's the latest hypothesis known as the response to injury theory. In other words, atherosclerosis is thought to be our body's response to an injury that occurs. And this information I'm going through now, that I'm going to start, is related to the article that I've also put on Blackboard on atherosclerosis. Now, I'd recommend you read that article. It shouldn't take you more than about half an hour or 45 minutes to read, but it'll really help clarify some of the information I'm going to go through. Some of the figures that I'm going to get, some of the graphics are from that article. So you might want to at least download it just to look at the graphics. And it also includes some nice pictures of lipoproteins. So that article is basically written for the lay population. It was written a few years ago by one of the leading researchers in atherosclerosis, so that's why I chose it. And it was published in a journal called Scientific American, which tends to be a really high-ranking journal. It used to be years ago that individuals thought that atherosclerosis or heart disease was simply due to the accumulation of lipids on the inside part of an artery. And if enough of those lipids accumulated, that artery would become blocked and blood flow would cease. But in recent years, they've realized it's a lot more complicated than that. And they've come to realize that it actually has to do with our own immune system. And they've also realized that it's not just lipids accumulating on the outside of the arteries, but that the lipid actually accumulates within the arterial wall. And in order to do that, our immune system becomes involved. So here's a basic synopsis of the response to injury theory. Cells within the coronary artery are injured. Causes of the injury can include hyperlipidemia, so high blood lipids, smoking, or hypertension, high blood pressure. Those are some of the main sources of injury. The main players involved in this process are cells of the immune system, but then also lipid material, most commonly LDL cholesterol. And here are the steps in the process. The first step is an injury to a coronary artery. The second step is macrophage accumulation. Third step is fatty streak formation. Then we have plaque progression. Then we have plaque rupture. So these are the basic items. What I'd like for you to be able to do is, for example, if I asked you to identify each of these steps and then briefly describe each of the steps, that's kind of what we want. I'd like for you guys to be able to link each one of these steps together so that you kind of understand the sequence of events that's related to atherosclerosis. So let's go through this. Then I'm going to use my laser pointer again, so hopefully you can see that. Now, kind of a busy slide, but what we're going to do on this slide is we're going to kind of go up left to right. We have injury here, number one, then we're going to look at macrophage accumulation, and then we're going to look at fatty streak formation. So this is kind of the first three steps of atherosclerosis that are shown on this slide. If we look over at the right, upper right, we have the heart. And if we're looking at a more close-up view of a specific area within the heart, we'd be looking at a coronary artery. So if we look at a close-up view of that and a cross-section of a coronary artery, we're now looking at this portion right over here on the slide. The inside of the artery or the blood channel is where the blood would be flowing in a normal situation. Now we have a couple different layers to this artery. We have the intima, which is the layer that's actually exposed to the blood flow. Beneath that we have the media, which is composed of a few different cell types, but overall mostly smooth muscle cells. And now the outer layers are called the adventitia. So this is a normal scenario. This is what an individual would like to have in order for blood to flow. Now if we come to the little bit below that, off to the left over here, we have LDL particles in the blood. So now what we're looking at is, here would be the blood channel, or basically the lumen of an artery, and here are the tissues surrounding that lumen. So we're going to look at the details here and go left to right, and as we go left to right here, that's going to correspond to our numbers here over on the right part of the slide to say 1, 2, and 3. Okay, so we're going to progress through this. A little bit more about the anatomy. Here is the blood channel. Now, lining that blood vessel or blood channel is a layer of endothelial cells. We looked at those before when we were talking about lipoprotein lipase, for example. Just below those endothelial cells is our layer of connective tissue that is a combination of cells, but mostly like connective tissue proteins in here. Then we have kind of an elastic tissue that's kind of a layer, and below that in the media we have mostly smooth muscle cells. Now if we start upper left, we have LDL particles here, these kind of orange circles. So LDL particles are delivering cholesterol to tissues. So they're normally part of the blood, and those LDL particles actually can penetrate through these layers of tissue as they deliver cholesterol to tissue. So it's not uncommon to find LDL particles in these outer layers here. Now, as we move left to right on this, we see an arrow designating a modified LDL particle. Now, and that's shown by yellow. There are a few ways that LDL can be modified, and I mentioned before that one way to modify LDL is oxidation. For example, cigarette smoke may oxidize LDL particles. Another way that they can be modified is they can actually be glycated or bound to glucose. So for example, if an individual has high blood glucose levels, there is the opportunity for that glucose to actually bind to amino acids and lipids within these LDL particles and modify them. Okay? Cigarette smoke can oxidize the amino acids or the lipids within these LDL. Bottom line is we have a modified LDL particle. Now, let's talk about what happens. One of the first steps in the response to injury theory is an injury to endothelial cells. And we said one of the items could be hyperlipidemia. So for example, if an individual's endothelial cells are exposed to high levels of LDL cholesterol, that can actually cause an injury within these endothelial cells. Another thing we said was hypertension can actually cause an injury to these endothelial cells. So if there are high amounts of pressure within this artery because of high blood pressure, these endothelial cells can actually be injured. And the other item was smoking, I mentioned. Those oxidants in smoking can also damage endothelial cells. Any source of injury can cause those endothelial cells to respond. One of the first responses that will happen is those endothelial cells will start expressing molecules called adhesion molecules. You can see it's labeled here in the center of this diagram. Adhesion molecules here. Adhesion molecules can bind to molecules in the blood or cells in the blood called monocytes. Remember, the monocytes are part of the immune system. Those adhesion molecules can also bind to T cells, other cells of the immune system. So the endothelial cell has been damaged, it's expressing adhesion molecules, and now those adhesion molecules can bind to monocytes that are typically traveling in the bloodstream. And monocytes are typically travel through the bloodstream and they respond to cues in the system or stimuli in the system. So the monocytes in this case normally would pass right by these endothelial cells unless the endothelial cells are expressing adhesion molecule. That allows the monocytes to attach. It slows their movement across the endothelial cells and they attach so that they can actually attach and then kind of roll through the endothelial cells. so they can pass right between the endothelial cells, and now the monocyte is actually in the intima of this arterial wall. Another item or molecule that these endothelial cells are going to secrete in response to injury are chemokines. And chemokines are signaling proteins. These chemokines are also attracting monocytes. So as the endothelial cell is damaged, the endothelial cell basically sends signals called chemokines to the immune system so that monocytes are attracted. So the monocytes are attracted to those chemokines, and that also stimulates their movement into the arterial wall, shown here. When those monocytes enter into this wall and interact with chemokines, they differentiate into macrophage. So they're called macrophage because macro means large and phage means to eat. So macrophage are cells in the immune system that consume other particles or cells. So the monocytes have now turned into macrophage. They are now fully differentiated macrophages that are allowed that consume other molecules. Towards the center of our diagram, we see that. We see a macrophage here that is actually consuming LDL particles. And the way that it's actually binding to the LDL particles is through an LDL receptor, also known as a scavenger receptor. Normally what happens when a cell accumulates LDL particles is it will downregulate the number of LDL receptors on its surface. Macrophages don't do that. They will continue to make more and more LDL receptors so that they consume more and more LDL particles. Those macrophages will accumulate so many LDL particles that they actually become completely full of LDL or lipids. So the overaccumulation of LDL by macrophage causes them to become what are known as foam cells, shown over here on the right. The accumulation of foam cells actually cause kind of a lipid accumulation that's known as a fatty streak. And so that's where we are here. Now, in this tissue, we also have T cells. T cells have also been attracted to the area. The T cells are also becoming involved in this process. The T cells are actually releasing these little inflammatory mediators, you can see here. So the T cells are releasing these molecules that are also kind of stimulating this process. So within a fatty streak, we can have a combination of foam cells and T cells. And the foam cells are actually full of those lipids. They can accumulate so many foam cells that you can actually cause this kind of bulge or fatty streak. Okay, I think I got everything there. So let's move on. So now that fatty streak has basically began to form. And now the system kind of continues to accumulate lipids. Step 4 of this process is what's called plaque progression. And the plaque is basically the next step in a fatty streak where significant amounts of these foam cells have accumulated. T cells have accumulated in this fatty streak. And now some of the cells are releasing molecules. These mediator molecules, you can kind of see some of them. They're trying to show them with these little like blue squiggly lines here. So some of these cells within this fatty streak are actually releasing molecules that are signaling the migration of smooth muscle cells to move from the bottom of the fatty streak to the top. So muscle cells are moving on top. Now, those muscle cells, when they get on top, are going to form what's known as a fibrous cap. And the reason it's a fibrous cap is because these molecules are now releasing a protein called collagen. And collagen is an extracellular matrix protein. It's a fibrous protein that is made to hold cells together. So collagen is the most common protein in the body, and its function is to hold cells together. So these smooth muscle cells are releasing collagen so that they can stick to one another. The reason they're doing that is they are forming a fairly solid structure or cap above this lipid core. So that is how the plaque actually progresses. So this entire area is called a plaque, which typically means an area that's a combination of T cells and foam cells. And some of those cells have actually died and they have released their lipids. Now, this process, which is shown here in this little inset, and is shown as number 4 down here, this process can kind of continue until that artery is actually completely blocked off. So more and more lipids could accumulate, more and more smooth muscle cells could form a cap on top, and this entire artery could eventually become blocked. But that only happens a small percentage of the time. Only about 15% of the time does this plaque progression actually lead to occlusion or closing off of the artery. What tends to happen more often is the next step, and that's called plaque rupture. Now at this point, what's happening is, if we look over on the right, we have some of these cells that are actually within this fatty streak or lipid core start releasing an enzyme called collagenase. And as its name implies, collagenase function is to break down collagen. So these cells within the lipid core are now releasing an enzyme that allows them to break through this collagen. These cells are also expressing a protein called tissue factor. Now what's happening is the collagenase is broken through the fibrous cap, so now that the blood can penetrate into this lipid core. When the blood interacts with tissue factor, it stimulates blood clotting. Once some of those cells within the blood, called platelets, are activated, we have a clotting cascade that's activated, which you guys have probably studied in other classes. So in other words, the exposure of blood to these cells within the lipid core that have tissue factor starts clotting. And another word for clot is called a thrombus. Now what happens is we can have this blood clot that can actually grow to a mass that will actually block off the artery. So that is typically what happens, is the artery is blocked off because of a clot. And if that's a coronary artery, then you lose blood flow to the tissue that's beyond it, so that's what can cause the death of that cardiac tissue. Now sometimes this clot will stay in the same place. However, sometimes this clot, parts of this clot will actually break off and travel to other tissues. For example, if that clot goes to the brain and continues to clot and it blocks off an artery in the brain, then that can cause a stroke. Okay, so this is typically what happens. So now if you go through this, if we summarize this, so if we go back to this and we say step 1 is basically an injury to the endothelial cells or the intimal layer of the artery. And the second step is macrophage accumulation. And that's when the macrophages from the immune system are present because of the injury. The macrophages engulf LDL particles by an unregulated pathway using the scavenger receptors, which are basically unregulated LDL receptors. That causes them to accumulate lipids. And then in step 3 is the fatty streak formation, which is the accumulation of lipids by macrophages that causes them to become foam cells. The process perpetuates as more monocytes are recruited to the area and are activated to macrophages that also engulf lipids. The foam cells in the fatty streak in the arterial wall have formed the fatty streak, which is a visible lipid bulge. The fatty streak is a precursor to a structure called the fibrous plaque. And during plaque progression, so on the next slide, we have some of the cells within the streak begin synthesizing collagen, a fibrous protein that's part of the fibrous plaque. The process can continue until the blockage of the artery occurs. However, more than often, we have another step, which is plaque rupture. And that is when cells within the fatty streak begin synthesizing a collagenase enzyme, or a matrix degrading enzyme, that breaks through the cap, exposes the blood to tissue factor on the cells, and that leads to clot formation. All right. So, I think that covers everything there. And as I mentioned, the article should help you clarify some of those things. Now, how long does this take? This can take a long time to progress to this. Now, what's kind of scary, though, is plaque progression, or basically atherosclerosis, can actually start in teenagers. And it can take years to develop, but the first parts of atherosclerosis can develop early on, and then it's just kind of a gradual process. Now, obviously, lifestyle and things like that have an impact, but it does take quite a long time to go through this process in general. Now, we're going to now look at how diet and lipoproteins are involved in that process. Now, the therapy for reducing cardiovascular disease has been based on the goals of reducing the concentration of LDL cholesterol and increasing that of HDL cholesterol in the plasma or blood. The reduction of LDL cholesterol is expected to limit the uptake of cholesterol by cells in the fatty streak. So in other words, whatever we can do to decrease LDL cholesterol, we decrease the likelihood that that LDL cholesterol is going to be involved in this process. And in general, the less LDL cholesterol we have in the blood, the less likely it is going to be modified or altered, for example, by glycation or oxidation. So that's why LDL cholesterol is considered to be the bad cholesterol. Now, the good cholesterol is considered to be HDL, because an increase in HDL cholesterol is considered to be of value because its presence indicates an increased rate of removal of cholesterol from the walls of arteries. Now, the role of dietary fats and cholesterol, we're going to look at those. Now, both of these can affect cardiovascular disease development by modulating plasma lipoprotein concentrations. So in other words, how does diet, specifically dietary fat, affect lipoprotein metabolism? Dietary modification remains the cornerstone of cardiovascular disease prevention, at least it should be. In other words, if dietary modifications don't work to cause the optimal results, then sometimes medications, for example, or other alternatives are actually required. But it should start with diet. Now, why is this important to control lipoproteins? Well, because it is estimated that every 10% increase in LDL in the blood increases the risk of cardiovascular disease by 20 to 30%. So there's a strong correlation between those. Now, the average U.S. diet contains approximately 34 to 38% of calories from fats, and that's quite a bit. The ideal number, or kind of the recommended percentage there, is 30% or lower. And of that fraction, we typically want 10% or less of our calories to come from saturated fats, approximately 10% of our calories to come from polyunsaturated fats, and approximately 10% of our calories to come from monounsaturated fatty acids. Now, if you don't think that's a lot, or it's not going to have an effect, here's one of the main points I want to make. Both the amount and type of fats affect plasma lipoproteins. In other words, even if a person does not decrease the total amount of fat that they consume, if they substitute unsaturated fats for saturated fats, they can actually improve their health in some ways, sometimes. Now, what I think you guys should do is take this 30% and figure out how many grams or pounds of fat you're consuming a year. So let's say, for example, a person's consuming 30% of their calories from fat, and they're eating 2,000 calories a day. How many of those 2,000 calories are from fat? Then figure out how many grams of fat that is per day. Then multiply that out per year. So go grams of fat per day by 365. Convert that to kilograms. So in other words, you're going to get probably maybe 10,000 or something grams of fat for a year, or maybe 20,000. Convert that to kilograms by dividing it by 1,000. Now you have kilograms of fat consumed in a year. Now multiply that by 2.2, and that will give you pounds of fat per year. And I think you'll be maybe kind of shocked at how many pounds of fat a person eats in a year. The point is that dietary fat can have a significant impact on a risk of cardiovascular disease. And if you think about it, if you're eating pounds and pounds of fat a year, you can kind of see where it will have an effect. So I'd go through that exercise. Now let's look at dietary saturated fat first. Saturated fatty acids tend to increase LDL cholesterol. In other words, the more saturated fat in the diet, it's going to tend to increase blood LDL cholesterol. If the average U.S. saturated fatty acid intake is about 12% of energy, it is typically higher than recommended amounts. Remember, we want 10% or less. It's typically higher than that 10%. If you look at the fatty acid composition of a typical diet, palmitic acid, which is the C16-0, is the major saturated fatty acid in most diets. It tends to be more common than stearic acid, and that's more common than myristic and then lauric acids. So, the problem is palmitic and myristic have the greatest LDL cholesterol raising potential. In other words, the most common fatty acid, saturated fatty acid in our diet, tends to be the one that can be the most harmful. So that's kind of a problem. Now, as we mentioned before, the saturated fatty acids tend to be higher in animal products than they are in plant products. So palmitic acid, for example, is no exception. It tends to be a lot of times about twice as high in an animal product as it is in a plant source. So if we look at this though, how does this happen? How do saturated fats increase blood LDL cholesterol? Well, saturated fatty acids decrease LDL receptor mediated endocytosis by decreasing LDL receptor at the plasma membrane of cells. In other words, the more dietary saturated fatty acids, the more cells have a tendency to express less LDL receptor. Therefore, the LDL stays in the blood because there's not receptors on cells to absorb that LDL. Remember, it's the LDL in the blood that tends to be really problematic. The longer it's in the blood, the more of a problem it can be. Now, the saturated fatty acids tend to do this by a couple different mechanisms. Saturated fatty acids tend to decrease expression of LDL receptor mRNA. And it's also thought that saturated fatty acids may decrease the ability of LDL receptor to recycle. So you have less on the cell membrane. One exception to this, stearic acid does not increase LDL cholesterol. It's one of the only exceptions for a saturated fatty acid. And it's thought that the reason it doesn't is because stearic acid can be converted to oleic acid in the body. So stearic is C18-0. It can be converted to the omega-9 fatty acid, oleic acid. And so it's thought that that's why stearic acid does not have the ability to increase or the tendency to increase LDL cholesterol in the blood. All right, trans fatty acids. We've all heard how bad they are. One of the reasons is because of their effects on lipoprotein metabolism. The most common trans fatty acid in the diet is oleic acid, which is a C18-1 trans fatty acid. It's a principal trans fatty acid in the diet. Now, some trans fatty acids occur naturally. For example, they're in quite a few animal products. And we talked about this before, that bacteria within those animals, some of them actually synthesize trans fatty acids, and then it ends up in the tissue of those animals. For example, the fat in beef, milk, and cheese contains approximately 2 to 8% trans fatty acids. However, overall, most trans fatty acids in the diet are from industrial hydrogenation of foods and oils, the polyunsaturated overall, about 6% of our dietary fatty acids are trans fatty acids. And I think that's probably come down a little bit for a few reasons, but pretty significant number. One of the reasons why they're bad is because most studies show that trans fatty acids increase LDL cholesterol. Okay, so that's one of the reasons they've gotten so much bad press is they increase LDL cholesterol. Some studies have also shown that they decrease HDL cholesterol, and you would not want that decrease in HDL cholesterol. Now, for this information, go by my slides. Your text maybe says things a little bit differently, but I've evaluated the data a little bit differently than the text. So, for example, the text probably says that they do decrease HDL cholesterol. I'm not all that convinced that they decrease HDL cholesterol as well. So use my slides on this. Either way, though, although some of the effects are still controversial, for example, do they decrease HDL or not? Reducing their amounts is unanimous. In other words, no one's going around saying, yeah, trans fatty acids are good for your lipoprotein metabolism. Dietary monounsaturated fatty acids. Okay, so why should a person consume monounsaturated fatty acids instead of saturated fatty acids? Because many studies have shown that when substituted for saturated fatty acids, monos reduce blood cholesterol. Remember, substituted. So it's typically not enough for a person just to continue to eat high amounts of saturated fats, but to also incorporate monos. You kind of want to substitute those. So your calories are basically the same or fewer, but what you're doing is you're changing where your fatty acids are coming from, consuming more monos than saturated. However, their effects are still controversial. Monounsaturated fatty acids tend to decrease LDL cholesterol. Okay, so that's a good effect. However, their effect on HDL cholesterol is still really controversial. So we're not too sure about that. Overall, it appears that monounsaturated fatty acids do not decrease LDL cholesterol as much as polyunsaturated fatty acids. Okay, so polyunsaturated fatty acids may have a better tendency to decrease LDL cholesterol. Now, what are some good sources of monounsaturated fatty acids? For example, we talked about these before. Olive and canola oils are good sources of monounsaturated fatty acids. For example, olive oil is about 75% oleic acid. Now, oleic acid is known to increase the expression of the LDL receptor. Because there's more expression of LDL receptor, that tends to decrease the amount of LDL cholesterol in the blood, right? More receptors, more uptake, that decreases the blood levels of LDL cholesterol. All right. Dietary polyunsaturated fatty acids. Polyunsaturated fatty acids tend to decrease LDL cholesterol. Linoleic acid is the main polyunsaturated fatty acid in our diet, and it tends to reduce both LDL and HDL cholesterol. So we'd have a good effect and a not so good effect. So, for example, why doesn't a person switch all of their fat to polyunsaturated fatty acids? One reason is because they're also maybe decreasing their HDL cholesterol, and that would not be a good thing. Another factor here to think about is when I said that one thing that can determine or kind of stimulate the uptake of LDL molecules by macrophage is the modification of those LDL particles. The more unsaturated fatty acids that an LDL particle has within its phospholipid layer, the more likely those fatty acids are to becoming oxidized. So that would be one argument for maybe consuming monounsaturated fatty acids also, because they have less double bonds. Those double bonds are not as susceptible to oxidation to a fatty acid that would have a lot more double bonds. So that would be another reason why a person's dietary intake of fatty acids should kind of be a mixture of polys, monos, and even some saturated fats. All right, so not only do polyunsaturated fatty acids affect lipoprotein metabolism, but Omega-3 fatty acids, for example, those high in fish oils, also lower blood triglycerides, and they reduce the risk of cardiovascular disease. Some of their effects are independent of their effects on plasma lipoproteins. For example, omega-3s reduce clotting, and they can also lower blood pressure. Now, how would they do that? Well, one mechanism is what we talked about before as far as eicosanoids. And I told you then, I said, well, one of the reasons to consume omega-3s is because the production of eicosanoids from omega-3s tends to reduce clotting. And now if you go back to that, some of the steps in atherosclerosis, we see that one of the typical steps is towards the end where we do have the stimulation of blood clotting. So omega-3s might be able to reduce that clotting process. That's one of their protective effects. They can also lower blood pressure. Again, remember, some of the omega-3 derived eicosanoids lower blood pressure, which would decrease the risk of injury to endothelial cells, and so that could decrease the incidence of these atherosclerosis steps. So overall, omega-3s have quite a few benefits, and so we've talked about that quite a bit. Now, what about dietary cholesterol? Most studies have shown that dietary cholesterol has less potent effects than saturated fatty acids. In other words, if a person is trying to control their LDL cholesterol levels, it's extremely important that not only do they look at dietary cholesterol intake, but also saturated fatty acids. You know, I'm amazed on how many times an individual will go to a physician and they'll talk about how to decrease their dietary cholesterol, but sometimes their intake of dietary saturated fatty acids is not even mentioned, and it really needs to be. So that's what that means. Now, it is even more variable than the response to dietary fats. In other words, some individuals respond well to decreases in dietary cholesterol, others really don't. So there's high variation on whether or not a person's dietary cholesterol reduction is going to have a significant reduction on their plasma LDL cholesterol levels. However, the best approach is to kind of use those in combination. For example, the worst combination would be high dietary cholesterol and high saturated fatty acids. The best would be low dietary cholesterol and low intake of saturated fatty acids. For example, saturated fatty acids reduced to less than 7% of energy. Remember, we said 10% or lower was good. And then cholesterol to 200 mg per day can result in plasma cholesterol being reduced by 15 to 20%. And that's really significant. This typical recommendation for dietary cholesterol is 300 milligrams a day or lower. So that would be a pretty significantly low amount of dietary cholesterol. Now, why don't we just raise HDL? Because raising HDL is not that simple. So, for example, I'm not convinced that altering dietary fat intake, for example, if you tell me increasing consumption of omega-3 fatty acids raises HDL, I don't think the data really says that. I think if you say increasing monounsaturated fatty acids increases HDL, I don't believe that. Okay? It tends to be highly variable, and it's not that easy to do. But in general, for example, physical activity tends to raise HDL cholesterol. For example, some studies showed that 45 minutes a day of exercise 4 times a week can raise it by 5 milligrams per deciliter of blood. And that might not seem like much of an increase, but if you consider an average amount of HDL in the blood is about 50, that's a 10% change just by exercising a few times a week. Now, exercise is also highly variable. For example, exercise effects might be slightly different depending on the intensity they exercise, the duration of the exercise, the gender of the individual that's exercising. So there's quite a few factors here, but overall, physical activity tends to have some good effects on HDL. Another thing, weight loss, especially around the waist, that can also help maintain or raise HDL cholesterol. Some individuals on the Atkins diet, for example, used to wonder why, look, I'm consuming high amounts of dietary fat, but my blood lipids are actually getting better. I'd say more times than not, it's actually that decrease in body weight that's responsible for the good changes in blood lipids rather than the diet itself. So for example, it's the weight loss that's causing the beneficial effects in the plasma lipids rather than the consumption of all that dietary lipids. Now, moderate, I should underline and highlight and maybe bold here, moderate consumption of alcohol has also been shown to raise HDL cholesterol. So some studies have shown that, for example, small regular intake of alcohol can actually have the effect of increasing HDL cholesterol. And it tends to be the ethanol itself. A lot of press goes to red wine, but other types of alcoholic beverages can also cause those beneficial effects. Did I say moderate? Yeah, moderate. So if you're downtown on a Thursday night and you're getting a little bit out of control of the alcohol consumption I happen to run into, I'm not going to buy the fact that you're saying, yeah, well, I'm just trying to increase my HDL cholesterol. Because I would say, remember, it's moderate. Now, get up and call a cab. All right, so overall, I think that the point of this is diet has an effect on the atherosclerosis process, and a lot of that effect has to do with how it regulates the plasma lipoproteins as far as LDL and HDL molecules. So I think that covers everything that I want to say here. So I will sign off.